So our next speaker, so we will have our next speaker, Justin Ichida. He is a prominent stem cell biologist and neuroscientist dedicated to advancing treatment for neurodegenerative diseases. He's an associate professor of stem cell biology and regenerative medicine at the University of Southern California. In 2022, Dr. Ichida joined BioMarin Pharmaceutical as a VP and head of neuroscience research. He's also a co-founder of Acura STEM, a biotechnology company focused on developing therapies for neurodegenerative diseases. His research primarily involves creating in vitro models of human neurodegenerative diseases by converting patients' skin fibroblasts into disease-affected neuron cells, facilitating the discovery of potential therapies. Without further ado, Justin.
Thanks. Now, I think my conclusion from that is I have too many jobs, but it's really a pleasure to be here in what feels like the epicenter of ASO technology in the CNS, and I've certainly drank the Kool-Aid for this modality as well. So I'm going to talk about some, as you mentioned, I am the head of neuroscience research at BioMarin, whose mission really is to develop therapies for rare disease. Today, I'm going to talk about some research from our lab at USC, much along the lines of the Project Mosaic talk earlier, in seeing how we could use iPSCs and leverage them to maybe identify points of convergence in sporadic ALS, and also to better leverage the genetic information that might be out there to point us in the right direction. So this was a perfect setup the last several talks, but we definitely think of ALS as a landscape with many different etiologies of the diseases. We do have, some known genetic forms. They might be clustered around mutations that cause instability or misfolding in proteins like TAR-DBP or TDP43 that are already prone to misfold. On the other hand, you might have mutations in genes that affect the protein clearance mechanisms within neurons, which could also lead to aggregation of things like TDP43. And then even some cytoskeletal genes, which may encode proteins that underlie the protein clearance machinery and their ability to function. And I think it's a perfectly good and sound strategy, and maybe the best strategy, to go after these known causes of the disease, maybe with designer ASOs. I think there are many people, or some people, going after that, and I think that has a high chance of success in some cases. One of the challenges is that we have this unknown or sporadic sector of patients where, as you can see, it's the bulk of cases, and we don't yet know all of the genetic causes that might underlie those diseases. In fact, we don't even know that there are genetic causes of those diseases. And therefore, it's been really difficult to try to understand if things that we're developing for some of these rare forms or testing on models of the rare forms really would translate to a mixed population like you would see in a sporadic ALS clinical trial. And this is what we're trying to potentially use iPSCs for, is to understand what therapeutic targets might be out there. Are there any therapeutic targets out there that might target a point of convergence with all these forms of ALS? As I mentioned, the genetics is not well understood for 90% of ALS patients, but we know that we can make iPSCs from these patients, and if there are genetic drivers within those patients' DNA, they will be preserved in the iPSCs and the motor neurons we make from them. And if we turn them into motor neurons, they may drive disease. I think that was like the overall hypothesis. And if so, we might be able to learn about, what are the differences in different forms of ALS, but also what proteins or pathways could we target to reverse some of the pathology that affects all patients. What we did to start, it was pretty unknown, of course, that iPSCs or motor neurons in a dish that are maybe a month old would would ever be able to mimic what happens in a patient's motor neurons when they get ALS in midlife. Um, but we nevertheless tried it, and this is a big graph bar chart here, just showing the rate of death of neurons made from iPSCs from these different individuals. So on the left, the far left, there are about 9 white bars. Those are all motor neurons made from unaffected control individuals, and they had a hazard rate or rate of death that we sort of averaged out to be 1. And then the next set of bars, the brown bars, that's the hazard rate, the rate of death of neurons made from C9orf72 ALS FTD patients. And you can see they're uniformly higher than the average of the controls, which goes along the lines of their genetic lesion in C9orf72 potentially driving their faster motor neuron death. And then the next set of bars are motor neuron survival times from different sporadic or unknown ALS cases. And I think what was very surprising to us was that all of the lines that we tested showed a faster rate of death compared to controls. And so, and then on the far right, there are more sort of rare genetic forms of ALS, including mutations in TDP43, also having the same sort of phenotype in a dish. And so you could also see on the left that if you look at the TDP43 within those neurons, in the control samples, they're all predominantly nuclear. In the C9 and sporadic ALS cases, you see a lot of that TDP43 leaking out and being sort of deposited in the cytoplasm and leaving the nucleus. So this is essentially what one sees to some extent in postmortem tissue of ALS patients. And even though we didn't know that there would be genetic causes of any of the sporadic ALS cases, I think one of the simplest interpretations of this data is that there are likely genetic drivers of motor neuron disease in many of these, in all the ones that we tested. Maybe it's polygenic, and so it's been difficult to really pin down all of the variants that contribute. But this suggests that we may be able to use these lines to understand how motor neurons from different types of ALS patients could react to the same type of therapeutic perturbation. Not to beat a dead horse, I'm definitely preaching to the choir here, but one of the convergence points we may be able to target would be the TDP43 pathology. We like that as a target because TDP43, the gene encoding TDP43 can harbor mutations that have been shown to cause ALS, and this pathology forms in 95 to 98% of patients. But it's not, as Don eloquently introduced, it's not that simple to target the pathology, because you don't want to knock down the gene because it has an important function in splicing. And if you try to kind of overexpress it to kind of restore nuclear function, what's been seen in the literature is that slight overexpression can actually lead to more pathology. You sort of get the aggregation that you see here in the cytoplasm. So it's a tough nut to crack, I would say. But I think it's something that we're game to try. And so how do we sort of go after things that might address TDP43 pathology? It might act upstream of it. We decided to do a screen, a genome-wide screen, using lines from a TDP43 patient. So this is an ALS FTD family where they harbor a single mutation in TDP43. As you can see by this schematic, I think one of the theories in the field is that many of the mutations that cause ALS in this TDP43 protein occur in this prion-like domain that causes it to aggregate more than it normally does. And that could lead to the pathology that one sees. But even in cases where there aren't mutations in that gene, it's thought that things that lead to higher concentrations of TDP43, like deficiencies in protein clearance or other types of stress, could also lead to this type of aggregation. And we thought we could model this potentially in the iPSC neurons, as I showed you previously. And so Jasper, Ruben Sigler, and Sharon Tu in our lab started collaborating with Ken Kosick and Juliana Acosta Uribe, who had in Colombia found what I think is one of the largest TDP-43 ALS FTD families known to date. So they have a spectrum of different ALS FTD phenotypes, but they all have the same mutation in TDP-43. And we made iPSCs from that patient line. And what we did was we generated these spheroids of neurons from that patient or from controls. As you can see on the left, if we look at like the known genetic form of ALS, C9orf72 patient neurons, they have a fast loss of their motor neurons in a dish compared to controls. And similarly, the neurons from this TDP-43 ALS FTD patient have also exhibited fast decline. So this was kind of a phenotype that we thought we could search for genes on where if we, with an eye on maybe ASO, antisense therapies in the future, could we find genes that when suppressed could rescue this type of degeneration. And so Jasper set up a genome-wide screen using CRISPR technology. And so I think we had done chemical screens like this, 6 or 7 years ago. It probably took us, 5 months to screen 2,000 compounds and pathways. This one took us like 2 weeks to screen all of the genes in the genome. And so essentially what we were looking for was, every neuron got a different guide RNA that suppressed one gene. And then we were just looking for the surviving neurons at the end of the survival assay and to see which guide RNAs were most enriched in those. And that would suggest that the suppression of those genes promoted the survival of these neurons. So we found a lot of things that we had seen before, like PIC5. We had seen things that other people have found in the literature, like nuclear porin genes. We also found a gene called VAV2, which caught our eye because what we wanted to do was really triangulate this with human genetic data to see what things might really modify disease in humans. And so Michael Snyder's group had just recently published a paper where they essentially leveraged iPSC motor neurons and said, motor neurons are likely the causal cell that's affected in ALS. And so what we should do is really only concentrate, or one approach is really to concentrate on the motor neuron genes and the regulatory regions that regulate the motor neuron genes, and sort of eliminate all of the other variants that are just contributing noise or decreasing our power of genetic analysis in this already sort of rare disease. And when they did that, they found genes that were really enriched either in the gene themselves or in the enhancer region of the motor neuron gene, enriched in genes that were differential, enriched in SNPs that were differential between people that had ALS and controls. And the top gene that they found where there were either lower VAV2 expression or loss of function variants in the gene that was enriched in controls was VAV2. So what that meant was that regulatory regions of VAV2 were enriched in variants that lowered the gene in controls, and in the gene itself, there were loss of function mutations that were more often in controls than ALS patients. On the right is some data that they pulled from this Answer ALS collaboration, where they made 1,000 iPSC lines from patients, ALS patients, and then made motor neurons from them. What was interesting is that in the motor neurons from that study, they found that the motor neurons from patients where they had lower VAV2 expression had a later age of onset, tended to have a later age of onset. So this suggests that whatever is in the genome, if it causes lower VAV2 expression, they might have later age of ALS onset. So less VAV2 is good to protect against ALS, essentially. We also looked in some existing clinical data from Eddie Lee's group, who had essentially taken brains of FTD or FTD ALS patients at autopsy and sorted out the nuclei from the neurons and then stained them for TDP43. And so they could actually do the RNA-seq on nuclei that from neurons that either had TDP43 or didn't. And what we found was that in neurons that didn't have TDP43, they had much lower VAV2 expression, they tended to, than the TDP43 positive neurons. So this could be because TDP43 regulates VAV2, but in another published data set from Kevin Egan's group, when they knocked down TDP43 in iPSC neurons, they did not see a lowering of VAV2. So it's not regulated by TDP43. And so one interpretation of this is just that the TDP43 negative, loss of function neurons that survived the latest actually had lower VAV2 levels in the brain. So as I mentioned, we're really interested in this ASO approach for many reasons. I think it's a very rapid way to test a therapeutic target in patients. And there are some other benefits, like, you might not knock down VAV2 too much in peripheral tissues compared to a small molecule approach. So as a tool and as a test case, we started testing this ASO approach in vitro. So we made ASOs that suppress VAV2 by around 50%. And then we tested them to see if they could rescue the survival of C9 or 72 ALS motor neurons, about 6 different sporadic ALS lines, and the TDP43 patient line that we started with. And in all cases, treatment with the ASO dramatically improved the survival of these ALS patient neurons across these different forms of ALS. VAV2, we're still trying to find out exactly how this works, but it turns out that VAV2 can activate multiple sort of GTPases downstream of it. Some of the things, interestingly, that regulates is protein clearance, protein acetylation. And so there may be multiple ways in which this single target is activating protective pathways. We do know already from the graph on the right that each of these different downstream targets of VAV2, when they're suppressed, as you would happen when VAV2 is suppressed, they all can contribute to improved neuron survival. So again, it seems like a single target acting through a multi-pronged downstream protective mechanism. What happens when we look at TDP43? I think one of the things we're interested in is seeing if we are upstream of TDP43 pathology and remedy that, because there are dozens of genes like STAthmin2 that when TDP43 is lost from the nucleus also are lost as a result. And so what you'd really like to do is restore proper splicing for all of those targets. And so what you can see here in the case of the C9 ORF72 motor neuron, as we saw before, there's a lot of there's a lot of TDP43 out there in the cytoplasm, but treatment with the VAV2 ASO seems to restore that protein to the nucleus, which is really what you want. We're beginning to now look at STAthmin2, for example. As Don introduced in the last talk, there's a cryptic splicing event that occurs in the absence of TDP43 from the nucleus. And with suppression of VAV2, we can suppress that truncated STAthmin2, that cryptic exon event, and begin to restore STAthmin2 levels. And I think we needed some optimization to look further at that. But I think we're seeing trends in the right direction. we've gone on to see how this might work in an in vivo model. We made an ASO for the mouse that suppresses VAV2 by about 50%. And this is a mouse that overexpresses human TDP43, so very rapidly develops the loss of nuclear TDP43 and the cytoplasmic inclusions that you see in patients. They also start having gait impairment very rapidly. So you can see the black line is the increase in gait impairment in these TDP43 mice without the ASO treatment, and in blue is the treatment of the VAV2 ASO. You can see that's quite a bit lower, and the survival time, on average, they improve by about 50% survival, but some of the animals have lived much, much longer. And we're looking into that. We think they have better biodistribution than some of the other animals in the study for the ASO. So that, and then what we also see is that the number of motor neurons is preserved in the spinal cord when we suppress VAV2 in these mice. We also see that neurofilament from the serum starts reducing even in this early time point of the study. This is, of course, a really important biomarker that looks at axonal degeneration or neurodegeneration that we think will be important clinically. So that's one example of using the iPSCs. And then in just a couple of slides, I wanted to illustrate a very similar but slightly different way in which we're using the iPSC data. So in collaboration with Sai Zhang and John Cooper Knock, they have been looking at the genetics and these rare variants and how they relate to survival of ALS patients instead of likelihood of getting the disease. What's interesting is they found a variant next to a gene called CCDC146, and they predict from the enhancer map of motor neurons that this regulates CCDC146. But the interesting thing is that patients that have this one variant basically survive like that blue line, whereas the average survival of ALS patients is the red line. So they essentially go downhill extremely quickly. And CCDC146 is another hit that came up in our screen, so we looked at that further. What it turns out is that variant actually seems to regulate CCDC146 expression in motor neurons. And so if you look here, the variant that is in the patients that decline very quickly leads to a slight increase in the expression of CCDC146. It also leads to shorter neurites in the neurons made from those patients. If they correct that variant to the other variant, they get much longer neurites. And also this variant that's in the patients that decline quickly, that leads to higher CCDC146 expression. It leads to higher TDP43 aggregation in these cells. And so that suggests that potentially, increasing a variant that increases CCDC146 levels causes very rapid decline for ALS patients. What happens if you now suppress CCDC146? So we made motor neurons from patients, C9 patients and sporadic patients, and suppressed CCDC146 by 50% with an ASO. And that now rescues the survival of those lines similarly to the controls. And then if we use that same mouse model and suppress CCD146 in those mice, you can see that they live quite a bit longer than the negative control treated mice. So, in conclusion, I think that there is a lot of potential for using, familial and sporadic ALS induced neurons to mimic some of the key hallmarks of ALS. I think it's probably not mimicking what happens with, aging of protein clearance mechanisms in humans, but very good at maybe understanding some of the, root causes of pathology in ALS patients. And using a genome-wide screen and convergence with genetics, we found that these two targets might be highly neuroprotective, at least they are in model systems. And so we're looking forward to seeing if we could advance ASOs against those targets. a lot of this work, most of this work was done by Jasper Ruben Sigler and Sharon Tu in our labs, two great scientists. Some of the legacy people have also contributed to this, and our collaborators, Sai Zhang and John Kupernock, have been working together nicely with us on the CCDC 146 story. So thank you very much.